So welcome everyone for joining the very first 2021 MBI webinar. I'm Jolin Chen, I'm your host today. For those who used to see Michael Farrell in these meetings, Michael has happily retired, and but I will try to carry on his wisdom in these meetings. So our first meeting is online, but hopefully we will have opportunity to meet face-to-face later the year. So before we formally start, I wish to acknowledge the people of the Kulin Nations on those lands Monash University is positioned. I pay my respects to their elders, past and present. So before we formally start the meeting, a small housekeeping item. Please ask your questions if you have any questions for the speakers. So please do via the Q&A button at the bottom of the Zoom window. So these questions will be answered at the end of the speaker's presentation. So chat function has been disabled, so questions must be sent via the Q&A button. Without further ado, I would like to welcome our speakers today. So we have two excellent speakers, Dr. Justin Adams and Associate Professor Alistair Evans. So Dr. Adams is Senior Lecturer at the Department of Anatomy and Developmental Biology at Monash University. His current research is to tackle paleobiological and anatomical research questions on living and extinct mammal species in Australia. And his research involves using very advanced medical imaging modalities such as CT, MRI, micro-CT, and synchrotron analysis. So welcome, Justin. Associate Professor Evans is a biologist and paleontologist with the School of Biological Sciences at Monash University. He's also an ARC Future Fellow. So recently he has developed new 3D imaging methods to quantify the very complex biological shapes, which can be used to predict the diet of the fossil mammals and the dinosaurs. I'm absolutely excited to have two experts in this field and also at the home Monash to speak to us today for both their individual and the collaborative research. So now I hand over to Justin, then Al for their presentations. So Justin, I will stop my sharing screen and you can take over from here.
Excellent, thank you, Shalon. Hey, welcome everybody. I'd really like to thank Jalin and Marin and everyone at Monash Biomedical Imaging for giving us this opportunity to chat with you a bit about some of our research that Al and I have been conducting both together and individually over the years. What we're going to be talking about today is this emerging discipline of cyberanatomy. And I'm going to talk a little bit about applications to humans and marsupials across the research I've done, and then be handing it over to Al to talk about some of his applications and works that we've done together. First and foremost, pretty important to start talking about what cyberanatomy actually is. So I'll chat a little bit about the brief history of cyberanatomy, what it is to us, and how it's an emerging discipline that is progressively developing. I'll then talk a little bit about some of the cyberanatomical research that I've done into human evolution. It's obviously just a tip of the iceberg in terms of the larger discipline. I'll talk a little bit about marsupials, a section I've titled Pouches Really. Then I'll hand over to Al. Al is going to talk about predictive cyberanatomy and then move into speaking about cyberanatomy and its integration into cyberspace, ways in which cyberanatomy is impacting a larger area of education and research and the entire discipline community. So to get started, and I guess to define cyberanatomy, the best way to think about it is as the digital representation of anatomy. And so this is the digital revolution as it's been applied to our discipline. This is a photo of me in front of what was the first CT scanner that was ever purchased in the United States at the Mayo Clinic. I was wandering down the hallway at the Mayo a number of years ago, and this was pointed out to me, and I was like, wow, this is absolutely amazing. One thing that I learned in that trip, which I didn't appreciate or understand up until the time they told me, is that the original CT scanner, which was called the EMI or the EMI for short, derived a fair bit of its funding from EMI, the record company, which had made a lot of money off the Beatles. So there's kind of an interesting deep time connection there between rock music and CT scanning, which I appreciated. We've come a long way from the original CT scan imagery, which looked like this off the IMI, to what we now see across different imaging capture methods. These span from things like micro CT scans at very high resolution. This is a fossil mandible, all the way through CT scans with contrast materials in order to enhance soft tissue differentiation, MRI scans, and then advances in surface scanning technology, both full color and just high precision, which is allowing us to capture a lot of very fine anatomical detail without necessarily going through the process of doing CT or MRI. Now, collectively, all of this imaging is allowing us a lot of opportunities that we wouldn't historically have had. So some of the first work that was done with digitizing anatomy within, say, hard tissue work, particularly in the paleosciences, had to do with fairly simple things like, well, now we can finally do things like virtual reconstructions. We can take partial specimens, we can mirror image them, we can position them in space, we can do quite a bit in order to start restoring anatomy and analyzing and assessing it. It also opened up new things that we would have never looked at before. So this is the vestibular cochlear system. So the balance organ that's inside the inner ear. This is a region that we never really would have looked at for evaluating evolution of trends or behaviors, but is now a thing that's open to us. And it extends all the way through into really complex interfaces between hard tissue and soft tissue, like understanding skull dynamics and the evolution of the brain across humans and other apes. For soft tissue, it's meant a lot of new changes. So from the very basic level, where our dissection work, which used to be a one-stop event, where we would do dissections, record them as best we can, now we can fully digitize in full color and high precision and actually share and record that data, allowing us to access different levels of dissection, particularly for rare animals like this koala limb, in perpetuity. It also allows us to start looking at things like volumetric relationships or internal organizations of limbs without having to go through the process of dissection. So being able to digitally extract materials and virtually dissect animals that otherwise we can't necessarily cut into because of preservation or their rarity. But I think it's really important to emphasize that cyberanatomy isn't just about the data capture methods and the ways that we do imaging, but it's also about what this has opened up for us. And that is a whole new way of quantifying. Because up until the cyberanatomy revolution, we were quite limited in terms of the way that we could quantify anatomy and start to do comparative analyses. Most of this were planar relationships where we're doing caliper measurements or other kinds of things which worked quite well for hard tissues, but didn't really work very well for soft tissues. So moving out of linear measurements and into a digital environment, coupled with advances in computing and software, this has really opened up new ways where instead of boiling a skull down to a few linear measurements, we now have the ability through these three-dimensional renders to do very complex assessments of shapes and contours and physical relationships and start evaluating hypotheses like across a whole series of monkey skulls about things like the shape of the face and whether that's biomechanically driven or evolutionarily driven. It's really opened up whole new opportunities and avenues for us in terms of the questions we can ask and the questions we can answer. Now, in terms of the work that I've done in human evolution, and there's a number of areas in which this is sort of touched in the imaging sciences, recently, in the last year, my team and I announced the discovery of the earliest specimen of the genus Homo, dating to over 2 million years from a site called Dromolen in South Africa. And that was a specimen pictured here, which is the neurocranium, or the back of the skull housing the brain, from a roughly 4-year-old child. As you can imagine, the skull bones are incredibly fragile, which meant that in terms of reconstructing what was originally hundreds of these specimens, went through iterations of micro CT scanning, and surface scanning in order to directly reconstruct the original specimen, but then also to guide through virtual reconstruction in order to round out and then interpret the anatomy that we were seeing. what was critical about this individual and what allowed us to make these major research advances in terms of understanding the evolution of our own genus was being able to take that digital data and then put it into a comparative context against specimens that were far flung, in this case, a specimen from Java, to be able to interpret and understand the shapes that we were seeing, the significance of what we're seeing older than 2 million years. There's a time separation here of almost 1 million years between these specimens. And so we were able to do a lot of digital reconstruction, direct comparison, and then critically extract information about the shape of the underlying brain, which is how we were able to assess and determine that there was already neurocranial expansion, so expansion of the brain relative to body size, at the very base of our genus. Other work that we've done recently and published on within the last couple of years, 2019, was some work that we did on a completely different scale of imaging, which was microimaging, essentially. One of the things that we were interested in looking at, and this was based on some prior research, were recordings of breastfeeding signals within teeth, looking at trace element deposition to try to establish patterns of behavior. The reason why we were so interested in this from a larger scale beyond breastfeeding behavior itself is that breastfeeding behavior speaks quite a bit about patterns of maternal care, and maternal care tells us a lot about ancient animals that we can't observe. Everything from social structure and diet to ecology, reproductive rates, habitat use, etc. So what we ended up doing was a process called laser ablation mass spectrometry. This is allowing us to do direct sampling and imaging of the patterns of representation of trace elements within teeth. And to boil down what we're seeing across these patterns, what we were able to determine by looking at elements like barium and lithium and calcium was that early Australopithecines, so dating to over 2 million years ago, they were breastfeeding their offspring for about 9 months or so. And then at that point, they would begin weaning them, and weaning would largely be complete at about 1 year of age. And that isn't perhaps the most surprising thing that we would find across a human ancestor of that size. But what was really fascinating was the return to breastfeeding in subsequent years. And what we found was that there was cyclical returns to breastfeeding up to about the age of 5 or 6 years. Now, that's an unusual thing, but not unusual if you're an orangutan. And so this raised a lot of interesting questions around the evolution of early human behavior patterns, but particularly whether or not some of these early African humans in our direct evolutionary relationship may have had behaviors like living orangs, where orangs have to follow seasonal fruits. They struggle sometimes to feed their offspring, and they have to return to breastfeeding as a way of keeping their offspring alive. So I'm going to move from human evolution and talk about what happens when an American comes to Australia, which is you end up exploring the world of pouches. This wasn't something that I personally thought was going to be the direction of my career, but here I am. These are some images of some possums that my wife and I took care of during the lockdown. That was our coronavirus project, was rescue possums. If you're an American and you've not really had a lot of exposure to marsupials, typically all you really know is charts like this that are presented in basic biology textbooks, which always put a pairing between a placental mammal and a marsupial mammal, and invariably sort of lead us to the impression that all marsupials are doing is being sort of the poor gap-filling cousin to a placental animal that occupies a niche in ecosystems. And so this sort of pairing of animals really sort of influences very strongly in cyberanatomy how we end up approaching questions and the questions we ask. But unfortunately, what this ends up doing is oftentimes sort of leading us astray in terms of the unique ecology that might exist for any of these animals that isn't just boiling down a quoll to being a cat or an antechinus to being a mouse. What we started doing is going for the big guns, going immediately to thylacines. And that's because thylacines are this classic textbook example of convergence between placental and marsupial mammals. But of course, the question that comes up when we use terms like convergence in biology is, do we mean that morphologically? Do we mean it functionally? Do we mean it behaviorally? Do we mean it ecologically? Do we need it genetically? What is the level of convergence that we're really describing? And have we ever really assessed this to the point where we can say that the thylacine is just a pouched version of a wolf or a dog with stripes, which is this dominant perspective that frequently comes out in a lot of comparative research. It hasn't really helped that when we want to look at thylacines, we don't have a lot of living marsupials to directly compare them to. And in fact, the largest of the carnivorous marsupials that is still living, the Tasmanian Devil, which oftentimes shows up in these kinds of comparative studies, well, it's an animal that is separated from the thylacine by about 35 million years of independent evolution, which for context is the amount of evolutionary distance between an elephant seal and a house cat. My house cat does not appreciate this photo being shown in public, but there you go. Well, in order to start unpacking this, and again, some of the work that we've done through Monash Biomedical Imaging has been the groundbreaking work done by my PhD student, or former PhD student, now Dr. Douglas Ravinsky. Douglas did really incredible work tracking down thylacines for one, but also doing some incredible analyses involving looking at the shape of the skull, a lot of CT scanning, a lot of soft tissue work, and has come up with some really amazing outcomes. Now, one of those, and a thing that I think would surprise a lot of folks, was that we didn't really have an understanding of how large thylacines really were. There were no accurate historical records that were established, and so this last year in the Royal Society, we published a paper that used CT scanning, cross-sectional biomechanics, 3D scanning methods, convex hull generation, a lot of different methods, in order to virtually reconstruct thylacines based on a number of specimens and then reevaluate what their body mass estimates likely were. We found that they were about half the size that everyone was thinking that they were and the numbers that were commonly being published. We also reinforced a really important point that doesn't end up coming up very much, which is the bimodal distribution of body size. In other words, there was a strong separation between male and female body mass. And this tells us quite a bit about the behaviors, the mating strategies, the other kinds of things that thylacines were likely doing in the wild. Now, there's more work coming, but I'm not going to show that because it's about to come out. So what I'll say is that there's quite a bit more on convergence with thylacines with placental carnivores that we're going to be publishing here in short order. Now, not everything's about thylacines. We've been doing some really interesting work across a number of different marsupial groups that just keep getting cuter and cuter. This is a bandicoot. And the bandicoots that we're particularly interested at present are evaluating this recently extinct group of bandicoots, the pig-footed bandicoots. Now, pig-footed bandicoots are extremely unusual in their adaptations. They had really, really long, skinny forelimbs, and that contrasts with the pattern that we see across living bandicoots that live in less arid environments because they're really focused on digging. So this is very much a digger's forelimb. This is not a digger's forelimb. But what this forelimb was doing for this animal, whether this had to do with operating at high speed, whether this had to do with thermal adaptations, has never really been unpacked. So again, work that we've done through MBI and are continuing to add to was working with some of the only preserved tissues for this animal that exist anywhere in the world. There's only 10 of these that exist as spirit specimens at Museums Victoria, and doing digital reconstruction out of MRI scans to start establishing patterns of muscle development, muscle orientation, and developing functional interpretations of what these animals would have done before they went extinct. This is helping us to start unpack aspects of its adaptation. So this is just a brief chart that shows some muscle proportions between the extinct pig-footed bandicoot and a whole series of other bandicoots and other marsupials. Really shows consolidation of different muscle groups, a lot of technical details. But what this is allowing us to do is finally test the hypothesis around whether or not pig-footed bandicoots with these extremely bizarre forelimbs, whether they were adapted to high speed as part of being what we call a cursorial adaptation, where they run around at high speed, it's potentially a way of escaping from predatory animals, or whether or not the elongation that we see in their limbs is more like what we see across various animals that inhabit arid and semi-arid ecosystems, where limb elongation or extremity elongation is about a pattern of thermal adaptation, allowing the animal to conserve energy and locomote across a landscape that is of a particular type. So there's a lot that we're doing with that in the marsupial community, but I don't want to sort of end with this idea that Australian mammals are all marsupials. I somehow got sucked into the world of marine mammals. I still don't understand how that happened. But my about to graduate PhD student, James Rule, did manage to convince me to jump from marsupials into marine mammals for a while. I don't know if I'll stay there. But there was this amazing project and series of projects that James did over the last couple of years, looking at this transition which took place. In Australia, we're used to seeing fur seals or eared seals. We can see the distinctive ears here. But that hasn't always been the case, because historically there's been suggestions that the marine mammal fauna was dominated by true seals or earless seals, like our elephant seal here, and monk seals, and seals that we're used to seeing in Arctic and Antarctic environments. Well, James, again, CT scanning that we did through Monash Biomedical Imaging, really unlocked the secrets of a series of seal fossils that have been found in New Zealand, which James described over the last couple of years. These have led to really radical revisions to our understanding of seal anatomy and adaptation. One of the key parts of this has been flipping the narrative around where these seals evolved and distributed from. Prior hypotheses focused on northern Atlantic origins, whereas it's now been demonstrated by these fossil remains from New Zealand that the origin center for monk seals actually came from the southern hemisphere. And we've described the earliest of these through a series of publications, including new species in the northern hemisphere as well. James has done something very unusual as a PhD student. He's described several new species and genera of fossil seals, which has just been fantastic for him. And again, through scanning done at MBI, allowed us to do virtual reconstructions in order to establish patterns of their adaptations, how they were feeding, and where they fit into larger patterns of seal evolution. So with that said, I'm going to stop my timer, and I'm going to turn over to Al so that he can carry you through with the rest of our talk.
Great, thanks Justin, and thanks Justin, Marine and MBI for inviting us to tell you about some of our research in cyberanatomy. So I'm going to continue some of the themes that Justin has introduced by looking at predictive cyberanatomy to start off with. So here we're trying to use the technology of imaging and then using computer analysis to understand things that previously had been beyond our ken. So here I'm going to show you some new techniques that I and my collaborators have developed over the last few years to do things like quantify shape and complexity of morphological systems, and also using them to investigate new rules of biological development that can tell us about the evolution and development of animals and plants. So through these techniques, we are able to do a lot of different types of predictions. We can predict things like how aspects of humans have evolved over millions of years. We can predict what fossils that we haven't found would look like. Justin showed some of the fossils that he has found, but we can actually use some of these techniques to predict what undiscovered fossils look like. We can use these techniques to uncover the diets of extinct animals, some related to us and some not, and also look at some of these general rules of biological development to predict the development and evolution of animals and plants over hundreds of millions of years. The main system that I use in my research is the teeth of mammals. And here is just a very small spattering of the immense diversity of just modern mammals and their teeth. And teeth are used for many different things. They're basically the toolkit of mammals. And so having many different shapes tells us that they have many different functions in the life of the animal: capturing food, breaking food, sieving food, and defending themselves. So one of the things that has made me very interested in morphology is understanding the relationships between simple and complex. So how can we say that one object is more complex than another? And then what can we use for that information to understand animals and their biology? So what we're going to do here is take a tooth. This is a tooth of a cat. wild cat from Europe, but if you look in the back of your cat's mouth, you would find this same sort of V-shaped tooth. So we're looking down on the tooth, and it has this long blade that comes along here. We can 3D image this through CT, surface scanning, photogrammetry, and we can use some techniques like geographic information systems to analyze the shape of this tooth. So when we take this tooth, we can basically make an orientation map. So looking at every little point on the surface, pretend that it's a mountain range and say, this yellow bit at the top is facing north, this green bit is facing south, and we break up all of the surface into these different parts. We can then use the GIS algorithms to group any parts of the face that are, say, of the surface that are facing east here into one big patch. There's another big patch over here that is red. We count all of these and we get a measure of the complexity of the surface, which we call orientation patch count, or OPC. So this one tooth here has an OPC by this measure of 37. So what's the use of this? What we can do is use this to look at the tooth shape of a variety of mammals to understand how their tooth shape complexity is related to their diet. So we can look at two different main mammal groups, the carnivorans, which are members of the order Carnivora, which include carnivorous things like red fox and herbivorous things like giant pandas. And we can also look at rodents. And so there are rodents that are carnivores and rodents that are herbivores. So we can take the three-dimensional scans of these tooth rows from the back of a red fox. This looks a lot like a dog. And when we calculate the complexity of this tooth row, it comes out as 119 patches. When we look at the giant panda, it's more than twice the complexity. So when we move from a diet of meat to a diet of plants, we need more complex teeth to be able to break down the very strong and tough plant materials like bamboo. If we look at rodents, we find very different tooth shapes that have evolved independently, but we see very similar complexity of the water rat, which is invertebrates, aquatic invertebrates, to the red fox. And we see very similar complexity in the woolly rat that eats grass compared to the giant panda that eats bamboo. So basically what we have here is a measure of the diet of an animal, which is independent of the taxonomy of the type of mammal that it is. And so we can use this technique to predict the diets of extinct mammals. One of the main groups that we've investigated is a group of mammals called multituberculates. And the name multituberculates basically refers to their teeth that have lots of little bumps on them, tubercles on the surface. And these were the most successful group of mammals back along the age of dinosaurs, back in the Jurassic and the Cretaceous. So we know that mammals back then were very small, and there's been expectations that mammals were actually forced to be small, and they didn't really have the opportunity to grow large because the dinosaurs were taking all the good jobs. They were being the big carnivores and the big herbivores. So can we find out whether mammals were constrained as much by the dinosaurs as has been expected? So to do that, we can look at the 160 million year history of multituberculates and see how their tooth shapes have changed and what this tells us about their diet. So we have very early multituberculates that have quite simple teeth with an OPC of 84. We get other ones that have these big bladed teeth with a higher complexity. We have more that have started expanding the complexity of these back teeth here. And then we have these really big and highly complex teeth in these latest forms. By looking at the whole history of multituberculates, we can actually see that many of these more complex tooth shapes, which enable us to infer that they were eating plants, actually evolved alongside dinosaurs. So some of these mammals were actually starting to compete with the herbivorous dinosaurs that existed at the time. And this completely upends our interpretation of what mammals were able to do in the age of dinosaurs. One of the other things that we'd like to do with fossils is that fossils are often broken, fragmented, and missing parts, as we saw with some of the human fossils that Justin was talking about. So this is one of the very famous fossils found in Victoria. It is an animal called an Ostrobesphenid. I won't ask you how to pronounce that, but it's a very small mammal. You can see how small this jaw is compared to a human finger, and it's been found from about 120 million years ago. Now, despite decades of searching by Tom Rich at the Museums Victoria, he's only ever found lower teeth, he's never found upper teeth. How could we ever work out what those other teeth look like and what that might tell us about who they're related to and their biological function? So we can use 3D scans of modern teeth and other fossil teeth and use sophisticated statistical shape analysis of 3D geometric morphometrics and statistical tests like two-block partial least squares to look at the shape relationships between upper and lower teeth. Once we do this, we can see that when looking at many different species, we can see how the change in the shape of the lower teeth matches the shape of the upper teeth. And here we're looking from inside the mouth, and here we can look down on top of the teeth. So these are three-dimensional models that are basically predicting when we change the shape of the lower tooth, what the upper tooth looks like. And so using those statistical techniques, we can go back and take our fossil tooth and predict what the upper tooth would look like based on this statistical relationship. We can then test whether this really works by taking our fossil tooth, which is down the bottom here, and our predicted tooth at the top here, and see if they fit together, and so they would have been chewing food. And so when we do that, we can see, in fact, yes, they fit together really well. And so this is very likely a very strong prediction of what this fossil would look like, hopefully one day when we actually find it. The next thing I'm going to talk about is some general rules of anatomy that we can use in cyberanatomy to understand the evolution of lineages like humans. So humans, across our evolutionary history, our teeth have changed in size dramatically. We have animals like the Paranthropus here, which had large teeth. Australopithecus afarensis, like Justin was mentioning before, we looked at some of their teeth. They have smaller teeth, and modern humans have some of the smallest teeth in our lineage of hominins. Now, there's always been questions about why tooth sizes changed. Some of the questions that haven't really been answered is how these tooth sizes have changed. So we know we have some good ideas of why tooth sizes changed. We know that diet has changed over human history. Tool use has increased, and also cooking has become much more important. And all of these things have meant that we don't need as big teeth and jaws in order to break food. But how exactly have these tooth sizes been changed, and what determines the size of teeth in an embryo? We know that we still have some difficulties with the sizes of our teeth in modern humans. The third tooth back here, the third molar, we call the wisdom tooth, is the most common elective surgery carried out in hospitals because obviously there's some sort of mismatch between the size of teeth and the size of our jaws. So understanding how these mechanisms work may help us to alleviate these problems. So we tried to understand what exactly is happening in the development of teeth. And our understanding started with, if in the embryo we start with a single tooth, it creates a second tooth, and it actually inhibits the size of that second developing tooth, that molar, and then that inhibits the size of the third tooth coming after that. And so by manipulative experiments, we can see whether this model makes any sense. We do this in mice, using genetically engineered mice, where we can basically make the teeth glow with green fluorescent protein, and then track the sizes of teeth as we do particular experiments. And through those experiments, we found that there are some very simple relationships of the sizes of teeth along the row, where the teeth from front to back decrease in size linearly, they stay the same size, or they increase in size linearly like that. So we can see how these patterns work in modern humans. In modern humans, in babies up to the age of 6 or so, they have just these 5 teeth in the lower jaw at the front. We're going to look at these back teeth, which we call deciduous premolars. When you get to the age of about 8 or 10, you start growing new teeth at the back until you get this final wisdom tooth here. Now, if we look at thousands of individuals, we measured their size of their teeth, we can see that across these 5 teeth, we have a very strong linear pattern here and a very strong linear pattern here. So this, in fact, tells us that this pattern, the inhibitory cascade, is determining the size of teeth within modern humans. and we can do things like fit statistical models and show that only 0.02% of the variance is not explained by this model. And then we have a change from an increase to a decreased size in the middle of the tooth row. We can look at this same data by looking at 15 species of fossil hominins, and we can see that these patterns of linear increase and linear decrease are found throughout these animals. What this means is that when we have animals like Ardipithecus, an early hominin, whose second tooth here we've never found, if we use the predictions of the model, we can basically predict what the size of that tooth would be. Now, when we look at this data in more general terms, we can find some overall relationships of these patterns with size. And this enables us to predict the sizes of all the teeth from a single tooth. So if we find a single fossil tooth, hopefully looking just as nice as this, we can use the equations of the inhibitory cascade to predict the sizes of the rest of the teeth in the tooth row. One more example of a developmental rule that we've been working on very recently is looking at how individual teeth grow and how the shape of the tooth is formed. And so when teeth are made, they grow, they start not like a building, starting from the bottom going upwards, they start from the tip and they grow downwards. And so in this lion tooth, this is how a lion tooth forms. So basically, as the tooth gets longer, it expands laterally. So let's measure from any sort of 3D scans that we have of teeth, how this developmental process goes. So we can take the central axis of a tooth, we can measure cross sections through the teeth in the way that it grows, and then we can work out what the average radius of each of those cross sections is. When we do that, we get basically a plot that shows us the distance from the tip of the tooth and the radius of that tooth. And so normally plotting it, we get this red line here, which is close to a straight line. But if we take the log of both axes, we get a straight line. And that is a very straight line. Again, fitting a linear model to that gives an R-squared of 0.9998. Because this is looking at a linear pattern in log-log space, we can call this a power law function, a normal mathematical term for describing this type of relationship. And so because we're looking at a power law that's cascading down the tooth, we call this the power cascade model. So let's look at some other teeth to see whether they follow the same thing as the lion tooth does. We can look at saber-toothed cats. We can look at saber-toothed deer, elephant tusks, mammoth tusks, human canines. We can look at walruses, tusks, whales, bats, pigs, marsupials, all sorts of single cusp teeth that we have throughout mammals follow this general pattern. We can look at other vertebrates like bony fish. Here's a deep sea viperfish tooth, extinct giant sharks, extinct marine reptiles and snakes, extinct birds that had teeth. Back in the day, birds did have teeth. Dinosaurs, lots of other reptiles, all of these guys fit this model extremely well. So this seems to be telling us something very general about how teeth grow. When we have this type of cyberanatomical description of teeth, we can use it to simulate what teeth would look like. So in the grey is a real tooth, a real 3D model, and we can use the equations to simulate the shapes which are in orange. So all of these guys that we looked at before, all sorts of vertebrates, dinosaurs, mammals, and Babirusa pigs, we can show how these shapes have developed in the embryo. Not just in teeth, we can also find the same pattern in many other structures in animals. So we can see this linear pattern found in claws, mammals, dinosaurs, and birds. We can find it in hooves, which are obviously a modified claw in animals like springbok. We find it in horns, in dinosaurs, mammals, and birds, and turtles. The points of antlers, like an elk. The beaks of birds, and also in squid, which are obviously now getting into invertebrates. We find it in the fangs of spiders, and we even find it in the lemon tree and the rose bush in my backyard, the thorns that are growing on them. So what we seem to have here is a very general biological, perhaps law, the development of these pointed structures that's common throughout animals and plants. And so this must be related to basic biophysical properties of life and the way that these structures grow. And it gives us immense power to predict development and evolution of these animals and plants for hundreds of millions of years. So what I'm going to do now is look a bit more at what else cyberanatomy is able to do, and basically using cyberanatomy in a way that we would not be able to do in traditional anatomical studies. So a lot of the data that we have collected in many of our projects can now be released on online databases like Morphosource, run through Doug Boyer in Duke University. This is basically the GenBank of morphology, where we can put 3D scans of many different animals and structures there as a repository from scientific studies, or to enable sharing between researchers over from one side of the planet to the other. So an amazingly good resource that is bringing cyberanatomy to the scientific world. These new interface of Morphosource is incorporating things like live 3D views of the 3D models that are available in the database. So you can go and look at our horn that was scanned at MBI to see whether this is the type of structure that you want to download. We've got teeth and claws and many of the other things that that I've talked about here. The next thing that we can talk about is some great new online websites that are able to distribute three-dimensional and four-dimensional data through a simple web browser. So we can generate three-dimensional structures, any of the things that we've been talking about, in an interactive view on a website, and we can also make these into 4D animations. So the structures moving, chewing, limbs moving, etc. And we have used this in our online teaching for COVID starting last year, where because we couldn't get students into the lab to do the normal sorts of investigations that they would with the practical materials, we can put these online and students can look at them, answer questions, etc. And we've started using it for public outreach to show people the amazing aspects of anatomy of our animals. So these are some of the models that we've made: individual skulls, whole models, whole skeletons, and just a demonstration of what this looks like. If you go to Sketchfab now, you can see this CT scan that we made at MBI. We've changed the models and the materials so that the teeth look real and the bones look real. We've animated it. We've made annotations so that you can indicate where you want to, which is useful for teaching and for outreach, so that people can actually see the structures that you want them to see. We can use full-bodied skeletons like this kangaroo that was also scanned at MBI, placed in a somewhat anatomically accurate position in a tree, but we have all of the bones indicated by these annotations, and students can easily click through them and be shown exactly where all of these bones are in their true anatomical place. And this enables us to do things that are actually even difficult while you're got the skeleton in front of you in the lab. So this is the types of innovations that that COVID forced us into, but we will be using from now on. So at MBI, we've had some great fun of scanning some amazing things. This is a whale that's been put through the somatom at MBI. It is a baby pygmy whale, because you think, how could you possibly fit a whale in there? So it's the smallest of one of the most enigmatic whales that are found in the oceans. So hopefully we're giving you some great ideas about what cyberanatomy is and how it is a major renaissance in anatomy, looking at things that previously were impossible or very difficult. We're using these to look at our own animals in Australia, so bringing marsupials up to the same level as understanding of our placental mammals like us, and we're able to release this to the world through outreach and through scientific distribution. So thanks to so many of the museums, institutions, Monash and others around the world who have contributed to this work. Thanks very much.
Well, thanks, Al, and thanks, Justin. That was an excellent, excellent presentation. I thought human images are beautiful, but you bring it to the next level. The image is really, really amazing. So are there any questions from the audience? So I guess people are still typing in. I got a question for both of you, sort of from more fundamental imaging point of view. So you look at multiple imaging modalities, CT and sometimes optical images. Is there a need, just generally speaking, to bring multimodality images into one model? I guess that will bring challenges. Is there a need to do so? For example, Justin, you showed muscle images, function images from MRI, but skeleton could be better with CT. Then you could have both anatomical and functional model altogether.
Yeah, and you actually highlight a really important area of some active development in terms of trying to maximize what we can get from, say, soft tissue contrasts using advanced ways of either staining tissues or avoiding stained tissues by things like phase contrast imagery at the synchrotron. But yes, sometimes we do in fact attempt to co-register data between, say, MRI and micro CT or CT. It is complicated, as you'd appreciate, with some of the smaller marsupials, because the average marsupial size is actually not a kangaroo. We do have the ability to isolate them within, say, tubes or other containers, and that can make it more feasible to keep them in the same positions to allow for later co-registration of data. But it is a bit of a challenge, and we do oftentimes play around with those. But ultimately, yes, it's part of an area in which we're exploring new directions to try to get the most both for hard tissue and soft tissue simultaneously.
And even surface models. So we have some specimens like a thylacine done an MRI, a CT, and a surface scan. So you get the stripes, you get the bones, and you get the muscles and the brain.
Yeah. We have a question from Michael DeVere. So question for you, Al. So lovely talk to molar non-pointed teeth, followed by a morphological law.
Yes, they do. So we can look at many other tooth shapes that have not just single cusps, single pointed parts, but multiple parts. And all of those individual pointed parts follow the same rule. So we can look at the individual cusps on human molars and they all follow the same rule. So it seems to be a generic, this is the way in which you make a pointy bit. If you need to make more pointy bits, you just replicate them over the surface of the tooth. And we think it's the same thing for things like horns, antlers, spines, like many other structures.
Jolene, I've got a question. I don't seem to be able to type into the Q&A. Can I just ask it? Is that okay?
Go ahead, Gary.
Look, great talk, gentlemen. Really fascinating work. Justin, I was intrigued. You made a comment at one stage that one of your students has discovered new species, I think you said. Is that correct? And can you systematically use imaging to search and discover new species?
That's a great question, Gary. Certainly in paleontology, that's where James has been working, in the sense of taking fossil specimens that have never been looked at, never been described, digitally extracting content out of imaging, and then using the morphology that comes from that in order to make arguments around this fossil specimen represents new morphologies, new adaptations, new things, and therefore represents a new species, a new genus, something that's never been described before anywhere within the literature. And sometimes reinvestigating fossils, things that were originally called something, but by unlocking new aspects of anatomy using imaging, are able to demonstrate, no, there are actually fundamental differences of this animal to something else that we thought. I think the final part of that is with the soft tissue work. and a big thing that Al and I are working on heavily at this point, is unlocking a lot of historical data that was just never really looked at. A lot of marsupial species went extinct before they were really ever investigated by science, which means that we know bits and pieces about them because they were alive long enough to be put into a jar, but they weren't alive long enough to actually evaluate what they were doing. And so as we progressively develop a baseline understanding of what they are and what their adaptations are, that progressively pulls us towards being able to discover, well, this thing that they used to think was this, well, it probably isn't, because it's just not structured in a way that is like the animals it was originally identified as.
Great, thanks a lot, Justin. I have a question for Al.
Yep.
I'm curious to know whether you think we should be having a data repository like, I think, what was it called, a morphological data repository here at Monash, together with the computational facilities, because that data showed over in Duke. I had a look at it, and it's fascinating stuff. Do you think we should be setting that up here?
Well, I think it would be a good idea to have something like that. And I mean, I set one up 15 years ago that, it was a very big, difficult process. Duke and NSF have put in huge amounts of money to establish the morphosource. And it may be possible to basically make a distributed duplicate of the morphosource or something similar that we can run a lot of the morphological data through here. So yes, I mean, we would use such a thing. There's been a lot of discussions throughout Australia about funding this type of database infrastructure, but it hasn't been particularly successful yet.
To add on to that, Gary, and not to get us too lost in the weeds, I'm happy to chat with you about an initiative that a group of about 17 institutions put together last year that I was part of the proposed steering committee on in order to get that sort of thing going and funded. And there is a huge drive, as Al said, to find ways to make that linked with things like Atlas of Living Australia or other kinds of major repositories of information that would allow for the kinds of sharing that we're finding is just absolutely essential for us moving forward.
Great, thanks, Justin. Be happy to take that up with you offline.
That's very interesting. That's I guess the 3D database in a way is very useful, right? I was thinking whether it's possible to do 3D printing just on top of that, and then you get all sorts of toys, and it could be all right. Great.
It's not just about toys, but one of the first things that I did back in 2015 with the ready availability of high resolution full color surface scanners was develop a fossil repository. And what that's allowed us to do digitally is distribute data to people who normally wouldn't be able to travel to South Africa in order to look at specific fossil specimens. But it's also allowed us to start printing these things in high resolution and in full color and make those available to people to do study. One of the things we've done over the past year has been doing extensive 3D printing to build research collections here to fill the gap for our inability to travel internationally.
Yeah.
And we've been using these online databases to be able to help our students and other students research students who wouldn't be able to get to museums or to CT scanners, to be able to start projects that in the past would be impossible without being able to scan things yourself.
These little humans from South Africa that otherwise I would have to travel, for a day and a half in order to access are now just sitting on my desk.
Yeah, amazing. Yeah. So perhaps another question. Oh, there's one from the Q&A from Michael Devere. Do we have an idea why the true seals are no longer in Australia? Or are they? So he's not sure.
So it's a great question, Mike. In essence, there seems to have been a fairly substantial turnover event which took place. And these are things that do occur within both marine and terrestrial ecosystems. which largely are likely due to changing local conditions, things like shifting patterns in marine resources, changing patterns in overall oceanic distribution, so nearshore environments. And then sometimes what you find with turnover events are other animals are just a bit more successful in one way or another, and they end up edging out the game. Where fur seals may have been more optimized to local conditions, it makes it less pleasant for local populations to continue persisting, and they just locally go extinct. So, you know, there's a lot of reasons that can drive it. We capture parts of that within the fossil record, and then the hard work is in terms of trying to figure out the why and the how of the bits that we don't necessarily get in the record.
Great, thank you, Justin. I guess if there's no more question, let's bring an end to our session. Let's thank our excellent speakers again, Justin and Al. Thanks. Give you a virtual applause. So just before we go, we have our next seminar happening on 11th of March. So we have two speakers, Dr. Kamlesh Power and Dr. Shinjun Zhong, talk about AI in medical imaging. So welcome everyone to join that session down the track. Okay, thanks again. Thanks to everyone for attending this session, and see you next time.
Thank you.
Thank you.